Good afternoon. Thank you all for joining us today. And welcome to the third annual REEU Research Symposium. So we've been very fortunate to have this program growing for some time now, and each year it gets a little bit bigger. So our research symposium simply is an outgrowth of a year-long program that integrates academics, internships, and now dissemination of that information. So in helping undergraduates prepare for successful careers in the agricultural and natural resource sciences or in STEM careers, we hold this annual research symposia for all students in our Texas A&M REEU program, which is the Research and Extension Experiences for Undergraduates, a part of USDA's programming for education workforce development. Our program is called the Science Influencers, and today we have the good fortune of hearing from our current science influencers, but also from several other REEU students nationwide. And so we'll kick things off here in just a moment. I'm Gary Wingenbach, by the way. I'm one of the co-directors here for the REU program, but I'd also like to introduce our project director, Dr. Holly Leggett. Holly, do you have any words for our presenters?
Yes, thank y'all for joining us today. I'm excited to hear the presentations. I think I'm more excited to know also that we have other REU participants from around the country joining us. So this is an exciting day to not only hear about the projects that we've been shepherding our students through this past year, but also to hear about projects that we don't really know a whole lot about, as well as to have their mentors joining us. So this is exciting, and I'm excited to hear 13 great presentations.
Thank you, Dr. Leggett. And I also like to call upon Dr. Barbara Gastel. Please introduce yourself. Is Dr. Gastel?
One second. I think that, hold on. I think she needs to, we need to enable.
Yeah, now I'm enabled. Thank you. Thank you very much. And just want to say, glad to be here. Looking forward to the presentations. I know our science influencers here have been doing great work, and look forward to learning about the great work of our guests.
Fantastic. Thank you, Dr. Gastel. And welcome again to everyone who is joining us near and far. And to get things started off this afternoon, we're going to start with Alexandra Ash. Alexandra was a participant in the Science Influencers REU group, but also in another REU program. And she will tell us all about her summer research experience. Alexandra?
All right. Okay. I have everything set up, I hope. All right. Can everybody hear me fine? I'm just checking. Yes? Okay. Cool. So, hi, everybody. I'm going to share real quick. And out. There we go. Let me put my view. Perfect. Well, hi, good afternoon, everybody. And I'm here to welcome you guys also to the Science Influencer Symposium for 2024. My name is Alexandra Ash, and I'm a biology major at Texas A&M International University, which is in South Texas, right on the border to Mexico. So quite a little bit far away from College Station, but still somewhat close by. And it's been it's also been a very busy year at the Science Influencer Program. And I've learned a whole lot about science communication and the importance of sharing that scientific knowledge to the public. So I'd like to take a quick moment to thank Dr. Leggett, Wingenbach, and Dr. Gastel as well for giving me that opportunity to participate in this program. I really think more people should participate in it because I've learned a whole lot. So thank you guys. And so I'm going to tell you about the internship that I did this past summer. This past summer, I had the chance to conduct research at Texas A&M in the Department of Plant Pathology and Microbiology. I worked for Dr. Shim and two incredibly talented graduate students to develop a project titled The Bioremediation of Sargassum Using Antifungal Bacteria. Now, for a bit of background, sargassum is a brown algae that floats on the ocean surface, and it forms aggregations when it accumulates too much. So these floating aggregations prevent the growth of seagrass and corals by blocking sunlight, since the seaweed creates a sort of cover over the ocean surface. And then these large masses of seaweed get washed ashore and block shorelines. And this not only looks terrible, it's very unsightly, but it also smells horrible. If you've seen these seaweed aggregations, the smell is something akin to like rotten egg. It's that bad. And yeah, so this smell is caused by the seaweed aggregations catching pollutants from the water, and it sort of stays on the surface there, and it leaches those pollutants into the water, and then that's what causes that terrible rotten egg smell, which is not what you want to smell when you're on vacation at the beach. And speaking of, hope you guys can see that, but this is a photo that my mom took actually when we were on vacation to Punta Maroma, Quintana Roo, which is by Cancun, if you guys don't know. And so let me zoom in a little more here. But that, I think I can point here. That is a person for scale. So if you can see that here, that is a massive seaweed pile. It's like taller than this person up here. I'm like moving this a bit too much. I hope you guys aren't getting dizzy. But yeah, so my main objective of this project was to test whether sargassum could be used to promote the growth of antifungal bacteria. And specifically, I wanted to test each of the bacteria strains. I called the 3 bacteria strains A, B, and C. I wanted to test their resistance to FOV4, which is a very prevalent and devastating fungal pathogen that affects cotton fields. And when I say it affects cotton fields, that's an understatement. FOV4 completely decimates cotton fields. So it's a huge problem in the agricultural sector. And that's what I want to focus on in this project too. It's a way to bridge not only conservation with this sargassum, but also it bridges conservation, microbiology, and agriculture in one multidisciplinary project. So I'm going to go over my methods real quick here. First, I amplified the 16S rRNA region of each bacteria strain, so for A, B, and C. And then I performed a gel extraction so I could obtain the DNA concentration through an instrument called a nanodrop. That's up here. And then we sent the DNA samples off for sequencing and for identification. And once we were able to identify the bacteria's relative taxonomy, we were able to determine the proper media for the growth, which was PDA or potato dextrose agar. This medium is great for both bacteria and fungal growth, which is why it was a good media to use. It wasn't our first choice. The ideal media would have been ISP2, but given the resources that we had, we stuck with PDA media. And so then we got those dried sargassum samples, we grinded them down with a mortar and pestle, and then we added different concentrations of it to the other media ingredients. We autoclaved it, and then we poured it into all these different Petri plates, which is shown up here in this figure. Wait. Yeah, that figure there. And as you can see here, the concentrations of the seaweed are 0.01, 0.1, 0.5, and 1%. So those are the different concentrations that we use there. So after that, I did a co-inoculation. So I pipetted FOV4 into the center of the plate and a bacteria strain on either side. And I used water and regular PDA media for the negative control of this project. So I've just been throwing a ton of methods at you. So you, after all this testing, what are the takeaways? Like, what did we find out from this project? Well, we learned that bacteria strains A and B showed the strongest antifungal activity. So that's these two right here. And you could tell that these are strong antifungal activities because of these inhibition zones right here. There's spaces where the FOV4 isn't growing. And if you'll see over here, this is our strain C that showed no antifungal resistance. The FOV4 was proliferating and growing and took over the whole plate here. So bacteria C had pretty much no antifungal properties. And what you'll notice in these graphs here is that there's a slight declining trend when it comes to seaweed concentration and antifungal bacteria growth. So these findings were the actual complete opposite of what we expected to happen. We wanted to see an upward trend because we wanted the seaweed to positively affect the growth of antifungal bacteria, but we saw a very consistent declining trend. And so that was a very unexpected result, but an interesting one nonetheless, since it was the complete opposite of what we were expecting. And another unexpected result were the bleaching properties of sargassum, which were present in the 0.5 and 1% seaweed concentrations. So as you can see here, FOV4, you see the fungi growing in the center there, it has a very rosy color in the low concentrations and no concentrations of seaweed. But as we move from left to right, we see that it kind of loses its rosy color here. And so that's why we say it has a sort of bleaching property. That's a placeholder word because it's not exactly bleaching. We don't know what it is or what part of the sargassum is causing this bleaching property. That could also be a very interesting avenue of research to pursue. And so my personal theory, I would think that it has something to do with the chemical composition of sargassum, since the chemical composition of sargassum has drastically changed since the 1980s, and it's slowly become more toxic over time. So overall, I got very unforeseen results, but that just made me ask more questions. Like, what would happen in higher seaweed concentrations? What would happen if we try a different media or a different species of seaweed? What would we observe then? So there you have it. I learned a lot from this project but mainly the importance of patience and troubleshooting, because there was a point in my methods actually that I couldn't get my gel electrophoresis bands to show properly. And that took about a week of troubleshooting because I was like, oh my God, what is happening? I'm following these procedures exactly, yet stuff still keeps going wrong. And I think that was the number one lesson I learned. Stuff goes wrong and you have to try a lot of different things to fix it and see what's up. So yeah, the root of that problem ended up being was that it was a faulty polymerase and we just ordered new ones. And then things started going smoothly after that. And I want to continue conducting research in the future. And this internship gave me a great framework and a ton of skills to send me on my way for that. So I'm very grateful that I got the chance to do this. And right now, that is it from me.
Thank you
If
so
you guys
much.
have any questions, I will be
Yes.
happy to answer them.
Yes, thank you so much, Alexandra. I do want to mention to everybody that the Q&A button is open. You can place your questions in there for presenters as they're presenting. We'll try to get that to everybody. We are on a very tight schedule this afternoon, so also you can post your questions, and if we don't get to them, we can always send them to the presenters, and I'm sure they'd be happy to respond. So round of applause for Alexandra. Very nice. Thank you, Alexandra. You can also use your emoticons in the reaction buttons at the top. There you go. Well done. And next, we are going to hear from Ava Tanaka. and Takanaka, pardon me. And Ava is going to be telling us all about a summer in Spain developing a biosensor for differential stroke diagnosis. Ava, can you hear us?
Wait, hi, this is my first time using Teams, so bear with
Great.
me. I don't know, can y'all see this?
Yes. Yes, we can.
Okay, we're all good?
Perfect. Perfect.
Okay, perfect. Hi, everybody. I'm Ava Takanaka, and I'm going to be talking to you guys about my summer that was spent developing. Okay, this title is a mouthful. Immunoelectroanalysis for differential stroke diagnosis, which I was basically working on a biosensor for stroke diagnosis, and this was the project that I was working on specifically at the lab. So I'm going to just start off with an overview of some internship details. So I was in Oviedo, Spain, which is a really small city in the north of Spain, at the University of Oviedo. And the professor I was working for was Dr. Teresa Fernandez, and she was so amazing. She's in this picture in the bottom left corner down here. And then the PhD student I was working with, his name was Pablo, and he was so awesome in teaching me everything that I need to know. The photos on here at the bottom are from the lab. You can kind of see the lab here, and these are some paper-based electrodes that I was working with. So I just wanted to throw that in because that was a big focus of my work this summer. These are some photos I have. I thought they would be fun to include just so you can see kind of where I was and what it was like. On the far left, this is a picture of a little beach town that was about a 30-minute bus ride from Oviedo. And I would just go there after work if I had time or on weekends, and I would go sit by the water and explore that little city. The middle is a picture of Oviedo from kind of the center of the downtown. And the bottom left of that picture, that's the Central Park they had, which was just beautiful, like the biggest trees you've ever seen. Like I would sit there and read and drink coffee, and when I had like time, and then this, I just thought it was pretty. And then this is the cathedral that's in the historic downtown. So yeah. Getting into my research, so the overall idea of the lab was to develop a biosensor for differential stroke diagnosis. So what they were going to do is take a patient sample, like a little COVID nose swab, plop it in the device, and it would flow through and be able to tell whether they had an ischemic or hemorrhagic stroke. So my work was focused on the vertical flow, optimizing vertical flow, doing electroanalysis, immunoassays, all for that protein detection. So as you can see, my project was broken down into 3 parts, and it has big scary names, but it really was, it's understandable once we get into it more. So the first thing I did was work on the immunoassays, which are like ELISAs. That's what they're called. So I performed 2 ELISAs, one with blocking buffer and one with washing buffer. So basically, the difference between these 2 solutions is the presence of bovine serum albumin, BSA, which we wanted to look at the impact that would have on protein detection because that's a protein. Also, I can't see the Teams page, so if anyone has questions, just leave them in the chat. But yeah, I was testing which would work better. And we ended up learning that the washing buffer worked just as well, if not better, than the blocking buffer, which had BSA in it. So we just went ahead and continued doing experiments using the washing buffer just so that there would be no errors in protein detection, so that when the device is really being used, there is accurate protein content coming from the patient and not being like messed up. The immunoassay wouldn't mess anything up. Then I moved on to testing the electrodes. So in the far left, these images here, I don't know if you can see my cursor, but those are literally paper strips that I was testing. I did some flow tests on those. So the top one here, these lines were drawn on with a hydrophobic pen, and this is literally melted wax crayons, and this is water. So I dropped water on it to see what would stop the flow better, and it ended up being the wax crayons, because when it's time to use the real paper-based electrode, we didn't want any of the solution to damage the like electrical components, so the device wouldn't be able to be used. So wax crayons worked best for this experiment. And then I performed some electrochemical testing, which is what cyclic voltammetry is, on screen printed electrodes and paper-based electrodes. Screen printed electrodes are just like plastic electrodes like this big, and then paper-based is the exact same thing, just made with paper. And basically, I was just testing to see how they would work in comparison here. Evidently, they both work the same. And honestly, the paper-based electrodes were even more sensitive when I was doing electrochemical testing, which is awesome because we want it to be able to be sensitive to proteins and we want it to work and all of these things. So this experiment went well. Paper-based electrodes work well for the device. So yeah, that was my electrochemical testing. I also did vertical flow optimization. So I was working on the device. Ideally, a sample will be dropped in here and it'll flow through the various layers of the device. The immunoassay will be completed and then it'll be able to, like the results will be read on like a little mobile device, ideally. So with this, I was messing around with sample pad and absorbent pad thicknesses to see what would work better for flow and just timing. And I have some results here. These were on screen printed electrodes, the results. And I believe the best ones from my memory were thick at 75 microliters of volume. And then I think a thin thick. Let me see here. Yeah, so these ones were the ones that worked best. We didn't want it to go, oh yeah, a thin, thick. Thick, thin, thick. We didn't want them to go too fast. The ones on this right here, the results in red and orange, the timing of that was too fast and we wanted the sample to be properly read, which is why here, these lines of pink and purple worked best. And then I performed the same testing on the paper-based electrodes and there were multiple layer combinations that worked well, which is good. So honestly, when the time comes to build the final device, my lab will have 4 different combinations of what worked well and what they can use. So that was the main focus of my research. I also want to mention, I didn't put this in the slides, I got to spend 2 weeks at the local hospital taking real patient samples so that when the time comes when they have their finalized device, they have real patient samples. So I was following around neurologists and neurosurgeons, watching them take patient samples, interacting with real patients who had stroke. And it was really amazing to see the people that we were building a medical device to help. And it really gave me a well-rounded perspective on my summer and on the work I was doing and how important biomedical engineering is. Lastly, I'll touch on my personal experience. So I was in Europe all summer, and with that, I did some traveling on the weekends. Yeah, personally, I really enjoyed the travel. The solo travel was amazing. I grew a lot in my independence because I did everything alone. I was taking planes, taxis, booking hotels. I was just wandering around alone. And it was a really interesting experience. Definitely got over a lot of anxiety in doing that. So this is a picture of Oviedo. This is some from, we have Madrid, Barcelona, Mallorca, Paris. These 2 pictures on the far right are actually from the hospital that I spent time at, which was awesome. Yeah, it was an amazing experience. And I think another big takeaway I got was to take risks. This summer, I sacrificed a lot of time and it was a risk to go at, 19 to another country to do research, but it was amazing. I think it was really good for my career, for my professional and personal growth. And it's something that I wouldn't change for the world. And I added new cultures. I loved being immersed in Spanish culture and learning some of their language and trying the foods and just how they live and work. I thought it was really interesting and it's different from American culture and it's something that I really grew to love and appreciate. So yes, I think this was my last slide, but I do want to just add on. I have a slide for Q&A, but I'll look at the questions after. I wanted to just say thank you, especially to the Science Influencers program. This summer would not have been possible without the belief of the professors here and without the funding and their teaching me on how to communicate and how to share. My life was truly changed and I wouldn't be able to do this without this program. So I just wanted to express my gratitude to you guys and thank you for that. And thank you guys so much for listening.
Thank you, Ava. We really appreciated hearing about it. And of course, Spain looks like a fantastic place to be. So again, congratulations to Ava. If you have questions, please post those up into the Q&A box and we'll get to those as we move along. So next up, we have an outside story. Brandon is going to help us learn something. Brandon's from Cal Poly State University, and he's going to help us learn something about carrot pomace supplementation on broiler performance. Brandon, are you ready to go?
Yes, I am.
All right. Flip on your. There we go. Got it.
Got it.
Thank you.
Sorry, give me 1 second. I need to pull it up. Hi, everybody. My name is Brandon Ng. I am a third-year animal science major at Cal Poly San Luis Obispo, and today I'm going to be talking about my research topic I did with Dr. King, which is focused on carrot pomace supplementation on broiler performance. Before we began our research in the summer, Dr. King took us across the country to visit many different processing facilities. In the winter, he took us to processing facilities all across California, more focused on poultry processing, which is why we went to Foster Farms Turkey, Foster Farms Chicken, and we also went to Superior Farms, which supplies every single Walmart with lamb and goat. And during the summer, we focused more on research facilities where we traveled to North Carolina and we went to visit Morris Associates, which helps create the technology in order for us to properly chill birds after processing. Went to North Carolina State University, where we met with the head of the poultry science department, and we also visit their Center of Marine Sciences and Technology, while also going to Fowl Resource International, they discovered an enzyme that is able to break down chicken feathers and create feed. These are some of the pictures of places we went to. We went to Superior Farms in Dixon, California, Foster Farms in Livingston, and Foster Farms in Turlock. And this was in North Carolina. The objective and purpose of our research this summer was to determine the effects of dietary carrot pomace on growth performance, feed intake, and conversion ratio of broiler chickens. Some background, because typical poultry diet in the industry consists of corn and soybean meal, but as inflation continues goes up and rising costs, along with competition for resources, the industry is constantly looking for new alternatives that, one, are more sustainable and are more cost efficient. That brings us to our topic, carrot pomace. Carrot pomace is a byproduct created from carrot juicing, and out of the many million tons of carrots that are juiced, 30 to 50% of the leftovers is produced as carrot pomace, which is rich in bioactive compounds such as carotenoids, while also being high in fiber and low in protein. Some of the methods we used for our research was, first, we obtained a frozen carrot pomace from Grimway Family Farms, and in order to process it, we first freeze-dried the carrot pomace at negative 20 degrees Celsius at 6.67 pascals for 24 hours before grinding it in a commercial spice grinder that was passed through a 20 mesh sieve with a diameter of 0.88 millimeters. For our birds that we used for our research, we used 360 one-day-old unsexed Ross 708 broiler chicks. How we set up our research was, we had 24 pens, each with 1.2 millimeters squared of area, with 15 birds in each, and we separated their feeds in 0%, which was their control, 4%, and 8%, which are the different percentages of carrot pomace we incorporated into their feed. And we also used a 3-phase feeding program over the 6 weeks that we grew the birds, with the first 2 weeks starting with the starter, and then a grower following with the finisher. And as you can see in these pictures, you can see the difference in the color based on the different percentages of carrot pomace we have. So what were the results of our research over the summer? So unfortunately, we did not come to a conclusion and that there was no significant effect of dietary carrot pomace on body weight and body weight gain. However, though, we did notice that in week 5 and 6, broiler feed diets that were fed 8% carrot pomace have reduced body weight and body weight gain, as you can see from the diagrams below. These are more diagrams. For figure 3, we show the feed intake over the weeks that we weighed the birds. And for figure 4, it shows the feed conversion ratio of the birds. And feed conversion ratio is how much pounds of food you need to put in order to produce 1 pound of weight gain. So in conclusion, we found that carrot pomace is high in fiber and low in protein. And this ties with the results we had with the broilers that were fed 8% carrot pomace, because since they were fed such high concentrations of carrot pomace, which was high in fiber, they're not able to maintain and fully incorporate all their feed, but instead are constantly were losing weight due to the high amounts of fiber. And we also found that 4% dietary inclusion rates produced the best results, which were they gained the most weight while eating the least amount of feed. And we concluded that in the future, if we were to continue researching on this topic, we should look for optimal inclusion ranges to properly find out what's the perfect amount of carrot pomace to efficiently and resourcefully boost the growth of birds, while also considering the cost of carrot pomace in broiler diets. Some of the additional experiences that we were able to enjoy thanks to Dr. King was aquaculture and algae growth lecture by Dr. Sarah Kumahara, who taught us the diverse characteristics and applications of algae, along with some of her other research topics, which were microbubbles and man-made self-sustaining ecosystems. And along with that, we were trained by Dr. Suraj Pokharo to become HACCP certified specialists. And HACCP stands for Hazard Analysis Critical Control Points. And the point of this is to figure out issues in processing that could pose potential hazards, identify them, and create a system to help not only prevent them, but also how to fix them in the case that the hazard does come up. As you can see in this picture, that's all of us with all our certificates after we finish the class. Some key takeaways from this research sermon was our cow poly model is learn by doing. And I feel that by being hands-on and actually raising the birds from day one to processing, you cannot learn better than by raising these chickens with your own 2 hands. And as a LA City boy, I didn't have any of these opportunities growing up. So this is a very great opportunity for me to not only learn more about the poultry industry, but also what it takes to truly raise a bird from day one to something that you get on the dinner table. And another great thing was we got to travel across the country. I've never been to North Carolina before, but thanks to Dr. King, I got to visit many different places that I thought I would never have been. And for acknowledgements, I'd like to thank Dr. King and Dr. Bennett for providing us with a once-in-a-lifetime opportunity, Dr. Kumahara and Dr. Pokharo for educating us, and I would like to thank all of you for your attention and engagement during this presentation. If you have any questions, feel free to ask, and that's it. Thank you, guys.
Thank you, Brandon. I'm sure I'm speaking for a few others when I say I love chicken,
I do too.
especially when it's roasted with some nice carrots. Awesome.
After the end of our research project, after we processed the birds, and we only used the breast for further testing to check for color and also to check for tenderness, but the rest of the birds we got to keep. So I got to take home a bunch of chicken and make some fried chicken for my friends. It was amazing. Some of the freshest chicken I ever had.
Very good. Very good. Again, thank you so much. And please post your questions in the Q&A button at the top of your screen. And next, we're going to hear from Davis Johnson. Davis spent some time this summer up in the northeast part of the country, and he's going to tell us all about it.
Yeah, howdy. Can y'all see my screen?
Yes.
With the presentation?
Yes.
Great. Yeah, so my name is Davis Johnson. I'm a sophomore biomedical engineer as well here at Texas A&M. And I had the opportunity to work with Dr. Maggie Chi and some really, really cool people at the MIT Chemical Engineering facility in Boston. So our lab primarily focused on microfluidics, and it is a way of modeling bodily functions in vitro or outside of the body. So a lot of different bodily functions are really, really hard and really expensive to model. And this is a simplified way of seeing that. And these pipelines, so these systems are mini pipelines, 200 micrometers across or 1/5 of a millimeter. And they're able to hold cells and such. And you're able to see how different cells interact, and that's what I studied. So this was my first time lab experience. Oops, sorry. This was my first time ever in a lab, so there was a lot of learning curves and learning basic procedures such as cell culture and learning different morphologies of cells. So throughout my time, I cultured 7 different cells. Um, Here they all are. Most of them are endothelial cells, so they're blood vessel cells, which is really, really cool because they're applicable not only to the retina, but to everywhere else in the body. And the next part that's really fundamental to microfluidics is immunostaining, which tags proteins. So you're able to see which proteins are expressed more. Um, so we had a code that once you stained for these proteins, in this picture, it's occludin, which signifies tight junction proteins, which is fundamental in keeping certain things out of the retina. So if we didn't have those proteins, which can degrade at times, we would see a lot more random, like, those floaters in your eyes, we would see a lot more of those. So being able to model the eye correctly and being able to quantitatively prove that is crucial. And then another area of study is chemical exposure. So another area of research is seeing at what point do these factors called COX-2 and other cytokines is what they're generally known as. At what point do they start affecting cells? So as you can see, CCL3, which is just a protein, really, really affects these endothelial cells at 100 nanograms per milliliter and is hardly significant before that. But these images are able to be quantified using a code that my graduate student made. And then all of this is turned into data eventually. So part of our lab was seeing how much infiltration through microfluidics chips we got by a certain type of cell. In this case, it's T cells, and the more infiltration means the more damage to the retina that would occur in certain situations. So I was in charge of making all of those data sets as well. And here's how we count the data. So we had 24 different images taken, each an hour apart, and we were able to run that through a code and then get percentages of infiltration. My project, though, was called the CoFlow project. It incorporates actually all of these simultaneously. Um, and the goal of CoFlow is to be able to seed multiple cell types in the same channel using different inlets. So this case is just dye. So it's 2 different dyes running through the same chip, running into the same channel. And the really, really cool part about this is since it's on a micrometer scale, laminar flow can be achieved really, really easily. So flows can be separated throughout the channel. It's a lot of fluid dynamics, but it's relatively simple to understand. But we were trying to be cost and eco-friendly. And so we would try to reuse these polymers and such. And so my biggest achievement was actually creating this solution that cleaned out the chip. So anytime that we ran these polymers through here, polymers also solidify in the chip. That was the goal of it. It would totally clog up the chip, and we added a dye so you could see it. So normally, it took 24 hours to partially clean out the chip, and it was really hard to see whether or not it worked. But after I created this solution, um, this is real time. It knocked almost all of it out in 5 seconds. But that was a lot of biochemistry that went into that as well. Um, And then one thing that I found really interesting was there's a lot of parallels to agriculture application. So the CoFlow process aims to be able to put two different cell types together, but letting them interact, but not mixing them. And that's something that's fundamentally found in all life sciences, including agriculture. So one area that I got to explore a lot and got to talk with a professional about was bioprinting meats. And that's one of the main focuses is trying to be able to print two different types of cells at the same time to achieve the same texture of meats. So it can be texturally and like tasting the same as traditional slaughtered meats. But yeah, this was a great preview of a ton of different routes I can take in college. And I learned how to become a collaborative researcher and how a real laboratory works. And then this CoFlow project could implement a different type of solution. Ultimately, we weren't able to get it to work with cells ever. That introduces a lot of factors because the fluid mechanics becomes really complicated whenever you bring into cells because each cell is uniquely shaped and they don't like to uniformly distribute themselves. But it's looking promising. So implementing more cell types into microfluidics chips could be the next step as well, trying out different cells. And then my biggest takeaways was how collaborative MIT was and supportive the culture was. And everyone in the lab group was a great mentor in some way to me. And yeah, thank you for the Science Influencers grant for funding me and the G Lab for hosting me.
Thank you, Davis. That was excellent. Again, if you have questions, please post those into the Q&A, and we'll try to get to those at the end of the presentations. So next up, we have Francesca, and we're going to hear about analyzing the effects of dietary fiber on primary and secondary kombucha processes. So, Francesca, I believe you.
Hello.
Hello. There we go. We see you. Very good.
Mm.
You can share your materials at the top of the screen. Click the share button.
Yeah, I'm gonna share it. Is it shared?
It's starting to come online. I think you might have to advance that. There you go. Perfect. We can
Okay.
see it.
Perfect. Well, good afternoon, everyone. My name is Francesca Caraballo Casiano. I'm from Puerto Rico, and I go to the University of Puerto Rico, Rio Piedras campus, majoring in nutrition and dietetics. So this past summer, I had the pleasure to do my SRP at UNL with the Crop to Food Innovation REEU program, and I work with Dr. Jennifer Ashton and Dr. Edward Dehan on the impact of dietary fiber on primary and secondary processes in kombucha. So to give a little background, kombucha is a fermented drink made with black or green tea, and we also has a symbiotic culture of bacteria and yeast, which contributes to the fermentation process that is also well known as the SCOBY. So the SCOBY primarily consists of a community of acetic acid bacteria and yeast. It also have lactic acid bacteria in it, and these organisms are potential probiotics that contribute to the health benefits of the drink. And also, we choose fiber because this has like one beneficial effect that is the effect on gut motility and prevention of constipation. And also data from NH shows so that the population in the US are 1/3 below the recommended level. So we think that supplementation could fill this gap, and that basically made our purpose. So we want to create a healthy drink that people will enjoy and with the dietary fiber, and they can choose better options without changing the mindset of the consumer. So we had two hypotheses. We had our primary hypothesis is that the addition of fiber for the kombucha will not alter the taste profile of it, so we can classify it as a high fiber food. And our second hypothesis was the addition of fiber to the primary fermentation process will alter the microbial composition of the SCOBY. So for our methods, I spent a lot of time in the kitchen lab, which was really exciting. I had a lot of fun in it, and I made the kombucha from scratch. So basically, what we did was boil the tea, and then we added on multiple jars, waited for it to cool. Then we add more water, and when we added the fiber and the sugar. So we use 5 different type of fiber. We use inulin, we use resistant maltodextrin, and fructooligosaccharides, and a 50-50 mix between inulin and FOS. And we also have 5 jars without the fiber on it that we're going to use on the secondary fermentation process. So after the 7 days of the fermentation of the drink, then we start the secondary fermentation process that we move those kombucha drink already made into the sealed bottle jar so we can have the carbonation process to it. And we also add a little bit of sugar and the flavors. So for those jars that didn't have the fiber in the beginning, we added in this secondary process. And also the fourth flavor that we used was blueberry, lavender and blueberry flavor, hibiscus, the normal kombucha flavor, and ginger. Then we also had some trial dilution and plating process to see the microbial growth in them. And as the results, we have 3 charts showing in this area that we can see how the pH level was going through the 7 days. We did about 2 or 3 rounds, but in these 2 graphs you can see the first round being lower than the second round, because when you are doing the kombucha drink, you use previously kombucha liquid. So it can help with the microbial growth more faster, but it depends on the pH range. So usually the pH range goes from 2.7 to 3.5, but if you use a really low pH drink before, it's gonna affect for your future rounds of kombucha. So that's why the first one was really low, because the previously liquid that we used was about 2 months fermentating, and then the second round was the liquid that we used was from the first round, so it was just 7 days of fermentation. So that's why it's higher, and you can see it lowering better. And the third chart is basically kombucha without glucose, because we also wanted to see how it was behaving and if we can do a kombucha low in glucose for future drinks. And in the flavor sensory panels that we did, we got to give it to my partners in the crop to food program to try it and give it me like rating. So we use a hedonic scale, which is basically 9 being most likely and 0 being less likely to, and we got to see which flavor was the best and which fiber. But the first sensory panel was about the flavor, and we only did the 5 flavor we had, and the most likely was the ginger one. And you can see here in the color, hibiscus was the most higher, but it was the lower in the flavor because it was really acidic in some way. And in the fiber sensory panel, we used that flavor that won. We used the ginger flavor and got to give it to my partners in the program again, but with the fibers on it. So the best fiber was the resistant maltodextrin. So for conclusions, we conclude that these results are significant for developing a high fiber drink tailored for consumers seeking health benefits, and the performance of the ginger flavors and the resistant maltodextrin fiber additive underscore the potential for creating this beverage. And these findings enable manufacturers to create a kombucha drink that satisfies consumer demand for tasty flavors and also offering a beneficial source of fiber for improved digestive health and well-being. And for future direction, we want to test whether there's microbial activity alters the fiber during the refrigeration shelf life, and also determine whether the addition of fiber to primary fermentation will alter the microbial composition of the organic acid profile of the kombucha. For me, this experience was I was like really grateful because it was my first time on the lab and my first research experience. And for me, being in Puerto Rico and moving to Nebraska for a whole 2 months alone was kind of scary, but really excited at the end. I really got to meet a lot of people. We were around 90 people in that program. And yeah, I had a lot of fun. I met a lot of people from grad school in the food science department. And also, I got to learn about this drink that I thought we didn't consume a lot at all in Puerto Rico. But I was talking to a registered dietitian that works in retail, and she told me there is actually a company in the west coast of Puerto Rico that sells kombucha, and it's called Kombucha Caribe. So, yeah. Thank you, everybody, for listening to me and having me here. And if you have any questions, let me know.
Thank you, Francesca. That was very interesting. All right, again, if you have questions, please post them up in the Q&A and we'll get to them as we can. So next up, we have Hope Bigby, and Hope is one of our science influencers here at Texas A&M University, and she'll be helping us understand something about the IFEDD Access to Care project. Hello, Hope.
Hi. Let me just go ahead and share my screen. Oh. Oh. My apologies. It's having me go into my settings. Here we go. Can y'all see my screen? Okay, great. So today I'm going to be talking about the major research component of my 2024 summer internship, the IFED Access to Care project. So just a little bit about me. I'm a nutrition undergraduate student here at Texas A&M. I'm actually graduating in December, and I will be attending Texas State for my master's in human nutrition. So first I wanted to introduce my mentor. Her name is Jessica Setnick. She is a registered dietitian, eating disorder specialist, speaker, speaking coach, researcher, and director of the International Federation of Eating Disorder Dietitians, which was what this research program was done under. And obviously she wears a lot of hats, but I was very lucky to be matched with her in the RD mentorship program. So we've actually been working together on this project since last September. And it was actually just last week that we submitted our research article for this project. So it's very exciting. So just some terms to define before we get started. MNT stands for Medical Nutrition Therapy and is defined as nutrition-based treatment provided by a registered dietitian. ED stands for eating disorder, RD stands for Registered Dietitian, and MAPIA is short for the Mental Health Parity and Addiction Equity Act, which is where we're going to start our background. So MAPIA was passed in 2008, and it was supposed to ensure parity in coverage, or coverage that is no more restrictive than for mental health/substance abuse benefits as for medical/surgical benefits. So this basically just means that there can't be additional restrictions or requirements to qualify for coverage just because care is for a mental health condition rather than a physical health condition. So an example of a violation of MAPIA, this is actually one that I pulled from one of our case studies, would be covering 26 visits with a dietitian for obesity, but only covering 10 for an eating disorder. And although the passage of MAPIA was supposed to ensure parity, it is widely known by eating disorder care professionals that coverage for MNT for eating disorder patients is routinely, improperly, and illegally denied. And whether it is intentional or not, these denials are illegal, and it's causing patients to not receive care that they are legally entitled to. When these improper denials happen, it can be an incredibly lengthy and time-consuming really high effort process to appeal them and restore coverage. For patients and their families, the eating disorder diagnosis is already difficult to deal with. They also have the job of securing other types of care for the eating disorder. And they also have normal lives. They have families to take care of, they're working, and they may not have time to go through the appeals process. For dietitians, they are going uncompensated or have delayed compensation for their normal work, but they're also spending additional unpaid hours working on appeals. And because if they do not have this additional time, they may be forced to choose not to accept insurance for payment, which shrinks care networks. And overall, this leads to patients having to pay out of pocket or discontinue care, which can lead to non-recovery of the eating disorder, relapse, hospitalization, ER visits, and death. So what is the Access to Care project? Our goal with this project was to capture the scope and scale of improper denial of insurance coverage for MNT for eating disorder patients, as well as identify significant features or trends. So these are kind of some of the questions that we were looking at. This was a pilot study, so we were just wanting to get an idea of what patients and providers are experiencing so that we can build a more in-depth and comprehensive survey later. So this project was started by Jessica Setnick as well as Domna Antoniadis. She is pictured here. She is a lawyer who very generously offered her legal expertise pro bono to IFED's dietitians to help them and their patients with their own disputes and appeals. But she and Jessica wanted to address the problem on a larger scale. So the work that I did on this project prior to the summer was a literature review and creating the data set. So for the literature review, I identified a total of 10 articles that were somewhat related to our topic. These are just some of the search terms there, but search revealed that this area has been relatively unstudied and no attempt has been made to capture this problem in the modern day and post the passage of MAPIA. So for data set creation, a message was sent out to dietitians via the IFED listserv. And we initially asked for three things: the name of the insurance company and insurance plan, the name of the employer and the state they're located in, and a narrative description of the problem that they've been having with insurance. And within 72 hours, we collected 172 cases. So these are all of the categories that I went through as I read each of the narratives and plugged in the information for each case. I believe they did end up being slightly different after Jessica went in and did a final review of the data, but this is pretty much what we were looking at: the plan types, the reason for denial process, and the types of interventions, which we'll talk about later. So as we're getting into the data analysis portion, I just wanted to go ahead and introduce Luella Fu and Gabrielle Salamanca. They are data analysts from San Francisco State University, and they were really key to this project. So they use Tableau Public and Google Sheets to create data visualizations and perform simple calculations to identify trends. So before I get into our findings, I just wanted to mention we had a really awesome opportunity to present our preliminary findings to insurance regulators and other related parties from the US government. And these images here are just a chart showing a patient's process of trying to get their denial appealed. And as you can see, it's a very long and ongoing and back and forth process.
So Hope, you only have about another minute and a half, so you'll need to hurry through the findings, please.
Okay. So our findings, violations are widespread throughout the country. They're not limited to a specific state or area of the country. We received cases from 26 of 50 states as well as DC. And then we found that it was an industry-wide problem and not limited to a certain company or group. And there was a total of 56 companies named in our study. So these are the 7 types of interventions that we found that were used in the cases. Some of these things like a reconsideration request, letters of medical necessity, peer-to-peer review, those are all things that people may not have time to do or are very effort intensive. So the primary outcomes from our cases, in only 7% of cases, they were successful in getting the improper denial reversed and getting their care covered for. In 15% of cases, patients had to pay out of pocket. In 13% of cases, they decided to discontinue care. And in 65% of cases, they went unresolved after months of waiting. So based on our data, the likelihood of getting an improper denial reversed was at most 50%, and that was with having 4 or more interventions. If you only had 3 interventions, there was only a 20% chance of insurance coverage, and if you only had 2, there's only a 16% chance of insurance coverage, which is very low. So just some discussion. These are just some graphics that I made over the summer about the prevalence of eating disorders, and just want to say such systemic illegal denial of care is unacceptable. And when we ignore this problem and allow the patients to get worse, it just causes them to need higher levels of care, which is more undue suffering for them and a higher expense and burden on the American healthcare system. So this just ties into
We're going to have to move on, Hope, so that we can make sure that everybody gets done before 2 hours. So we do appreciate you, and we thank you so much for the research that you presented.
Also, as a reminder that we have all of these materials posted on our site, and we'll be sharing that link here in just a second. So next up, we are going to hear from Jesus. Jesus is coming to us from Tarleton State University, and we're looking forward to his contribution. Jesus, are you ready?
I'm ready. I'm having problem with the camera. I'm gonna go over and just present because I have the pre-recorded video. So allow to share. Can you confirm it is presenting?
You are presenting. Yes.
Okay. So
You're good
I'm
to
gonna
go.
play it.
I don't see anything.
No?
Not yet.
Share screen. This. Oh my god. This window. For me, is presenting.
I don't see anything.
Jesus, can you go up to the share button in your Teams and share your screen with us?
Yeah, I'm doing it. I'm even pressing the window and the screen button, and it seems that it's presenting, and I'm looking back and forth, but apparently it's not working.
No, I tell you what, let's come back to you, all right?
Yeah, I
And
can go over the end. I'm so sorry.
that's all right.
Apologize.
I will try to look for it on my end as well. So then let's skip over to Caleb. And Caleb is from Texas A&M University, and he's going to discuss his findings from some deep cave this summer. Caleb, please take it away.
Hey. Let's see. Is it sharing at all?
I do not see it yet.
Hmm.
There we go.
Okay. Is it up now?
Yes.
Yes, we can see it.
Okay.
There.
Yeah, I'm gonna have to switch. Okay,
There you
does
go.
it all look good now?
Yes.
Okay, great. So, yeah, I am Keo Krubo. I'm a senior in biological and agricultural engineering at Texas A&M University. And my REEU experience was in cave microbiology and the different tools we used for researching that. So, Dr. Hazel Barton was my mentor, and I did the experience at University of Alabama. My field supervisor, who I did a lot of work with as well, was Max Kother. He was a PhD student in Dr. Hazel Barton's lab. As you can see, we're all expedition cavers, and our research revolves around cave microbiology. So what is cave microbiology, you might ask? Deep within caves, even miles, even in the dark, isolated places, there is a microbiome. And those microbes do produce different byproducts through their metabolic pathways. Some of those things are like different acids, and those are used to maybe dissolve the cave walls, and different things like that. So while studying it, we can discover things about microorganisms, speleogenesis, which is like the creation of cave formations or the dissolution of like limestone to create the cave. Or sometimes it's like antibiotic resistance or drug discovery and just different chemicals that we can use in biotechnology. So that's what Dr. Barton's lab deals with. So the main project that I helped with was with Max Kother, and we were looking at these massive caves in Borneo. I didn't get to go to Borneo, but we just worked with the samples from there. But as you can see on the right side, that picture, these caves are like maybe 300 feet wide. They're massive. You see a tiny person in that picture, and they're wondering how these were created because nothing really makes sense. And they think it's from microbiology processes. So mainly dealing with the nitrogen cycle. So there's birds and bats that fly into this cave, really deep in this cave. Their guano is a energy source for bacteria. And those bacteria are producing different reagents, different acids that are potentially dissolving the cave walls and like over a very long time have made these gigantic caves. We also did isotope ratio mass spectrometry to see the flow of nitrogen in the cave. And then we worked with some genomic extraction to see like how we could get the DNA to analyze out of this bird and bat guano because there's a lot of PCR inhibitors in there and it's just really hard to work with. So that's a little qubit that we used to prove that the DNA was readable. And yeah, there's me doing preparing samples. So I also get to help out with Asurus silva. We were researching cyanobacteria in caves, specifically lava tubes, to kind of understand how life might survive in lava tubes on another planet like Mars. So helped him with some environmental sampling. I did some growth media preparation, helped a few other projects with that as well. did some DNA extraction, and he showed me how he does sonication to extract the DNA. He had to work in this dark room with these green lights because cyanobacteria, they use light for energy, so you can't really have that messing with experiments. Next, I got to go to the National Speleological Society National Convention. There, it was all cave research and science presentations. There was a lot of networking. My mentor, Dr. Barton, actually gave one of the main talks at the event, and most of the other PhD students in the labs gave talks as well. But I learned an insane amount of cave science, and then also got to do some caving trips while we were there because it was in Alabama or Tennessee, and yeah, it was great. So, one of the main projects I also helped with, apart from all the grad students, was my own thing. I was developing a procedure to test these microscopic rope fibers because they've been dissolving in this cave. So they're wondering why, why they're dissolving. That's a really scary thing to have your ropes dissolve in the cave. It can be dangerous. So I figured out how to manage these little microscopic ropes, treat them, and then use a tensile testing machine to test if they were breaking easier than the non-treated ones. It was really cool to develop my own procedure, and it's going to be used in the project for years to come. For science communications, I also made these little Instagram reels, as you can see there, and they did really good. Got a lot of good feedback on them, and did some podcasts with all the grad students, which are on the YouTube channel. I'll have a QR code at the end if you want to check those out, and made a little survey like video on how to survey a cave with new technology. Other cave science, I was around cavers all the time, so they taught me how to do lidar analysis and actually search for caves using a computer and then go out and check them in person. Did some vertical techniques and also learned how to draft a cave map. So this is a cave I surveyed in Mexico, and they showed me how to draft it using Adobe Illustrator. In conclusion, the isotope ratio mass spectrometry showed good grouping. We could see how the nitrogen was flowing through the cave, gained new skills in cave science and microbiology, developed a nuanced method for genomic extraction from guano. We got really close on that. We got at least the DNA. It was proved that it was on its way to being extracted. They're still working on it. Developed a rope fiber testing procedure and gained a lot of experience in science communication. So I want to thank Dr. Barbara Gastel Wingenbach and Dr. Wingenbach and Dr. Leggett for the Science Communications program or Science Influencers program, and all the people I met at the University of Alabama that helped me along the way in different research and things like that, and my mentor, Dr. Barton. So that's it.
Excellent. Thank you so much. And it sounds like you had a wonderful experience. We certainly appreciate you sharing that with us today. All right, let's go to Luke Sprecher. Luke, I know you're in this list somewhere. And Luke is coming to us from Cal State University, LA.
Okay, can everyone see my screen?
Yes, we can.
Okay.
Go right ahead. Thank you.
Okay, so hi everyone. My name is Luke. My presentation is on the hydrogeologic study of the member basin aquifer system. And this is a transboundary multi-use aquifer. So it goes in between New Mexico and Mexico. And I'd like to point out my supervisor, Dr. Barry Hibbs, and co-supervisor, Dr. Jeffrey Rawling. And I was at the Cal State LA this summer, and I collected the data out in New Mexico. So a little bit about me. So I'm a junior at the University of Wisconsin-Madison. And then I work for the Center for Dairy Research at the CDR as an assistant cheesemaker. In my free time, I like to go out and play some disc golf. And then I'm also play Rocket League competitively for esports. And then so for the study area of the Members Basin, so this is an area that is a group of geohydrologic subbasins, and this covers about like 13,300 kilometers squared of the area, and this includes part of the U.S. and Mexico. So in the map here, the United States and then Mexico, and this represents a wide array of land use and land cover. So cities and towns in U.S. and Mexico, forests, rangeland, lowlands, and irrigated cropland, all things that are important to USDA, like administrative stuff. And then motivation. So this data like helps us provide new and updated groundwater data for like growing urban and existing agriculture, and this area is shared by the United States and Mexico. This is a 5-year study, so this uses major inorganic constituents and trace elements along with the environmental isotopes and the age dating of groundwater. And the purpose is to perform a hydrogeologic isotopic hydrochemical study of the transboundary membranous basin aquifer system. And this is a large binational aquifer system that lacks isotopic and specialized isotopic data on a regional scale. And this is a way to fill that data vacuum. And it supports the forest, rangeland, irrigated agriculture, and urban planning. And so several methods used to collect well samples. So there were city wells, which are the two on the bottom left, and then like irrigation wells and here, and then a solar well over here. So the parameters measured, so for the sake of time, the stuff that's highlighted in red is the stuff I'll be talking about, but we did index field parameters, general minerals, trace elements, and environmental isotopes. So I'll be talking about chloride, nitrate, total dissolved solids, lithium, and carbon-14. So for the total dissolved solids, if you look on the left, so this is data from January, and on the right is data from July of this year. If you look, so the standard drinking water quality is 500 milligrams per liter. And as you see, all the data from January that we got is under that. And we have 2 samples on the right from July that are over that. And then they all lie under the fresh water class. And then temperatures, if you look at the ones on the left side for January, the 2 are from like hot springs or like geosprings, which is why you would see the higher values there. And then for nitrate, chloride, and lithium, so everything seems to be good there. The nitrate standard is 10 milligrams per liter. All of the samples that we had were under that. Chloride was standard for the EPA, it was 250 milligrams per liter. All of that were under. Lithium is a little bit of concern as there's no specific value right now out for what's considered unhealthy for lithium. They just say about like above like 10 milligrams per liter. And for some of the samples you can see over here, that is above the 10 milligrams per liter. And then for age dating of groundwater, so this shows like how fast this aquifer is being replenished or how fast that groundwater is flowing through. So the smaller data or the smaller values show it's going through quicker, and the larger values are saying that it's been there a longer time. And this is a way to see how the groundwater is flowing, where parts are like recharging quicker versus parts that are recharging slower. And this is important to understand because this region of the US and Mexico, there's lots of nuclear waste that can go there. And you don't want to put your nuclear waste somewhere where the groundwater recharge is flowing quicker, because that can cause contamination problems versus somewhere if you don't put it where water is flowing, so it doesn't really contaminate the water as much. And then the research impact and importance. So it's important for the accurate data regarding well water composition and managing transboundary multi-use aquifers like the Members Basin. Water users in urban and agriculture areas in the Members Basin depend on this well water for water demands. And then this study is providing essential data for water users. And this is a year 1 baseline data, and this informs the next phase of the studies of the Members Basin Aquifer for the next 4 years. So there'll be 4 more students working on this project the next 4 years to carry on what I have here. And then the impact of this internship. So professional changes. From field experience, I was out there, got to acknowledge like what it's like out being in the field, collecting data, and also the use of using instruments and data collection like the multi-parameter sonde, which helped us get the pH, temperature, and other important things. And then personal changes, it helped me gain independence. So coming from Wisconsin, going to an area that's desert and not all like foresty, like it's different, like experience for sure, and it's way hotter. And then independence living on my own, getting that experience, and then application to career. So this laid the groundwork for my graduate studies in water management and like water quality, and getting understanding of kind of the groundwater and well water data, and being more like getting around that kind of information and working with it. And then collaboration and communication in team environments. So working as a team with other members, working together to get stuff done. And then I'd like to acknowledge USDA, NIFA, REU grant for giving me this opportunity and funding this, Dr. Barry Hibbs, Dr. Jeffrey Rowling, and Chris Cristales. Thank you.
All right. Thank you, Luke. Excellent. Thank you for sharing that. All right. Next up, we will attempt to, let me share my screen, and we'll attempt to watch our video from Jesus. Here we go.
Dr. Wingenbach, we can't hear him.
Say again.
There's no sound on my end.
Oh,
Me neither.
all right. That's not working so well. Let's move on to Mark, and then we will try to figure out the technical difficulties that we're having with the video. Mark?
Hello, can you hear me?
Yes.
Okay, let me share my screen. Um, okay. Let me see.
Should be a share screen button at the top of your panel.
Gotcha.
Very good. There we go.
Okay.
Just make that into the, there we go. Got it. Excellent.
Okay. Alrighty. So, hello, my name is Mark Villa, and today I'll be presenting my research on combined effect of light and heat on leaf functions. This study was part of the USDA REEU program, where I worked under the guidance of Dr. Christine Scafoni alongside grad students Eduardo Barragan and Carly Miller. A little about me. I'm passionate about fungi, and I'm currently studying biology at Cerritos College. This picture right here shows me holding a coral mushroom at the LA Mushroom Society festival at the LA Laboratorium. And these two photos, I also enjoy film photography. So I love taking photos of nature and stuff. And these two photos is just of moss with the sporophytes and then another mushroom. I'm not too sure which type of mushroom that is. Okay. Oh. Okay. The project was developed during my internship. I collaborated with Eduardo and Carly Miller right here. They're both right here. To collect plant samples from the California Botanical Garden. Our focus was to study how 5 native California species respond to combined stresses of heat and light. The objective of the research was to determine how light and heat combined impact leaf function in native plants, and to refine methods for studying heat stress in leaves, improving how we evaluate plant resilience to extreme conditions. And as you with the globe temperatures rising, they're rising about 1.17 degrees Celsius since the late 19th century. Plants are facing more extreme environmental conditions. In California, average temperatures have increased about 1.8 degrees Fahrenheit, with nighttime temperatures rising the fastest. This highlights the need to better understand how plants handle these changes. And here's a little video of the Earth warming up, but I'll let that play. It should be done soon. Yeah, and you can see it pick up around like the 70s with the temperatures increasing, and then from the 70s on, it just gets worse and worse. Okay, a little background for the research. Both light and heat stresses impact photosystem 2, a critical protein complex involved in photosynthesis. Stresses increase energy loss of stresses increases energy loss as heat or fluorescence, reducing efficiency. To measure plant health, we use the FE over FM ratio. A healthy plant scores around 0.82. I'm going to be going over that a little bit in the next slide. Yeah, to assess the heat sensitivity of plants and stuff, and the native species that we focused on, we calculated the T50, the temperature at which photosystem 2 loses 50% of its efficiency. This was determined using a water bath protocol by Krauss and others. This was the term allowing us to generate a thermosensitivity curve for each plant species. And on this one, this is the little curve that we're looking at and we focused on during the research. On the x-axis, it shows the temperatures, so 25 degrees Celsius and 45 degrees Celsius, and then our FE over FM ratios on the y-axis. And as you can see, it shows a little that the little line is that T50, the dotted red line. And on these photos, you could see it just shows like what the fluorescence meter uses. This is a little fluorescent meter right here on the top right. And the red being that it's healthy, the 0.82, all the red is, so it's accepting that photon and converting it into chemical energy. And then the blue spots right here are a little bit more less effective at doing that. And the five species we studied was the white sage, the Salvia apiana, mountain mahogany, chaparral whitethorn, desert apricot, and summer holly. And we select these species for the ecological importance and their ability to survive in California's diverse and often challenging environments. So we try to choose a little bit from different environments, so like the desert, the chaparral, locations and stuff like that. So our first goal was to study the combined effects of light and heat stresses on plant health. To do this, we exposed leaves to 8 to 10 temperature treatments under two conditions: low light, which is like lab light, like normal light, and high light, which we use the LED lights right here on the top left to simulate that the full sunlight. After 24 hours, we measured the recovery using the FE over FM ratio on the chlorophyll fluorometer, and we've calculated T50s for each condition. And then also our second goal was to refine methods for studying plant recovery after the heat stress. To achieve this, we tested 3 recovery approaches in a recovery in sealed bags with them closed up. Also recovery in open bags with a damp paper towel inside with all the leaves inside, and then recovery with the leaf petioles placed inside water. And then on these two photos, you can see that one with the leaf petioles placed in the water right there with all the temperatures marked. And the two species that we tested was the Cercocarpus and the Salvia. So I believe this one's the Salvia, and this one this is also the Salvia as well. I don't have a photo, but okay. And so our results in 4 out of the 5 species studied, the highlight significantly reduced heat tolerance, lowering T50 values by 1 to 6 degrees Celsius compared to low light. This suggests that studies conducted in dark conditions. So in the past, they wouldn't use any highlight; they would just use normal lab light. So with those studies, they may overestimate the heat tolerance of these plants because they're only focusing on the heat and not light as well. Desert apricot was the exception, showing no difference between light temperatures, and that's going to be right here on the bottom. This was the Prunus. The Prunus fermonti, which is the desert apricot, and that barely showed any difference between the low light and highlight between the confidence interval right here. And the rest, so the salvia, the rest of them, the summer holly and all of those did show a big difference. So on the on the y-axis, it shows the temperatures, 30, we did 30 to I believe 54 degrees Celsius, and then the FE over FM on the y-axis. And the white dots are the low light. Oh, sorry. Yeah, the white dots are the low light, and the black dots. Sorry, the black dots are the low light, and the white light dots are the highlight. And then let's keep going. So the results for the refining the methods, recovery methods generally had a little impact on the FE over FM values, except for mountain mahogany, which was the Cercocarpus. Open bag recovery led to worse. So what we found is the open bag recovery led to worse outcomes, likely due to hydration. This suggests the sealed bags or petioles placed in water are better options for avoiding the water loss. So on this one right here, on the x-axis is the FE over FM again.
1 minute, Mark. One.
Gotcha. The bag and the yellow is the bag, the orange is open, and the blue is water. Okay, and then a little discussion on the findings. Our findings show that the highlight significantly reduces heat tolerance in most species, with T50 values dropping by up to 6 degrees Celsius compared to low light conditions. This suggests studies done in dark and low light settings may overstimulate the plant's ability to handle heat stresses. The reduced tolerance under high light is likely caused by reactive oxygen species, which damage proteins and slow photosystem 2 repair. For recovery, we found that oxygen deprivation had a little effect, but dehydration, especially in open bags, negatively impacted recovery. Seal bags or petioles in water provided the best conditions for recovery, ensuring more accurate results. Overall, the study highlights the importance of examining combined stresses like heat and light and heat, and better understand plant responses in climate and just changing climate. And we would want to the implications for the future is just to see to have a higher temperature when recovering to stimulate more real world conditions. And from this internship, I gave me hands-on experience on both lab and field work. I've never done that in the past, so it gave me a chance to see how I liked it and see which one I liked a little bit more. It helped improve my research and public speaking skills and inspired me to pursue a career in assessing climate change.
We're gonna have to move on. Thank you.
No worries. Thank you.
Thank you, Mark. We appreciate that, hearing about that interesting research. And let's go with Jesus. Are you ready now? Do you have some slides ready?
I got the slides ready. I think now I'm sharing.
Well, I see our meeting room. There we go. Put that on
Yes.
presentation and we are good to go.
Thank
Excellent.
you. So, okay, confirm the camera is on. Yes, okay. Sorry for the inconvenience. My name is Jesus Cortez, and today I'm going to talk about my experience on this REEU summer program. So a little bit of my background. I'm from Puerto Rico. I'm a first generation graduate student, and my background mostly has been involved with horses. Most of them Paso Fino thoroughbred horses, which I'm been working with them from basic horsemanship through advanced techniques at veterinary medicine and research. I finished my bachelor's this past spring 2024, and this summer I got an opportunity to participate on this REEU program under Tarleton State University, which was an equine focused research. And after that program, I make the decision to start um, my master's degree at Tarleton State University with a long-term goal to go to vet school. So talking a little bit about the internship setting. So it was at Tarleton State University on Stephenville, Texas. Um, Tarleton is part of the A&M University system. And although it is a small campus, it has about 130 degree programs. Um, I was working under the mentorship of Dr. Jessica Leatherwood, a renowned equine nutritionist and physiologist. My experience working with her has been one of the most grateful experience that I've been have as a student and as a person because of the passion and professionalism that she shows and her graduate group. Um, so kind of talking a little bit about my personal development. So I think the first one that is most impacts me more was to adapt to this new environment. So this new culture, this new climate, and kind of being away for a long time from my home. It was the first time that I leave my home from such much time. Also increase my self confidence. So kind of push myself to speak in a different language that I was accustomed to talk every day. Meeting like-minded peoples, sharing our experiences, looking to a big a shared goal to look for our research and stuff. And also, since I moved to a new place, well, exploring new places, such as within the same Texas, we visit Amarillo, so Texas Tech, and at the end of that research program, we visit North Dakota, so we share with other folks their experiences too on this summer research program. On part of that professional development, so as my daily basis work, I improve my laboratory techniques such as aliquoting, sampling, laboratory safety, and um, assay preparations. As a professional and personal, horse handling management is something that I always look forward to improve as a horse owner, as a horse enthusiast. The problem solving and data interpretation is something as you are a researcher, you have ups and downs. So how resilient you are and how critical you are in order to overcome those problems is something that I really am working with it. And the part of pursue of that graduate career. So I start this fall. So that helps me also in that professional development. Now talking very briefly about what was the focus and goal with this project. So equine welfare has been a major concern in the last decades because of these high profile events such as the Kentucky Derby, futurity events, and Olympics has been affected for the late scratches and the reduction of participants because they've been injured or they present lameness, which is the first clinical sign that horses show us that there's a problem on their musculoskeletal system. So for us, in order to search a novel, non-invasive potential tool that we can detect lameness, electroencephalography has that access and cognition to welfare that we're looking for, and can improve that detection on those horses to prevent those injuries and lameness. We mainly focus on those low brainwave frequencies such as delta and theta. So these frequencies has been correlated with with pain, either the reduction and comfort of pain or the differentiation of that chronic pains that persons or other species can have. And so also we use a proof blood biomarker, which is prostaglandin E2, which is an anti-inflammatory mediator. We want to prove our objective and hypothesis, which is to investigate that relationship of a surface EEG and blood biomarker to lameness in mature horses. And our hypothesis was to that those low EEG frequencies will correlate with that established biomarker contingent on lameness scores. So those main results that we found, it was that PEG2, prostaglandin E2, has a moderate correlation with the EEG frequency delta, which is related to that pain decrease. or reduction. And also we found that theta, which is the one that is really correlated with chronic pain, has also a moderate correlation with the frequency, and at the same time, substance P has a moderate correlation, which is going to be explained further by my partner Sabrina. So we found that there's a main effect, an interaction within the lameness score that were assigned and with the frequencies. So we found that on non-lameness theta waves, there was a significant interaction and activity, which that biological activity needs further explanation, but we infer that those horses that were not lame are assessing that chronic pain that they could have because they've improved on a lameness evaluation, so they can at the same time decrease that pain that they could have. And for the biomarker, we found that there was no difference in time, but it was a difference on the concentration of a serum biomarker in comparison with plasma. So for further analysis, we're going to use serum plasma so we can have a concentration of more metabolites, so we can infer if they have more secretion of that eicosanoid. So our conclusion, this study highlights the potential relationship between the EEG wave frequency and pain presence in horses, particularly delta and theta bands, as they vary between lame and no-lameness categories. The investigation into blood biomarkers such as PG2 show moderate to though no significant correlations with surface EEG data, suggesting a possible avenue for early lameness detection. So we want to acknowledge Dr. Lewood's graduate and undergraduate students. We also want to acknowledge USDA and NIFA for their grants for this research program. So this is a reference, and thank you for your attention.
Thank you, Jesus. We really appreciate that. Again, all these materials will be online, and you can check them out in the Q&A box. I put in the link. So next up, Marisol Vasquez, another science influencer from Texas A&M International. And we're going to learn something about urban heat island. Marisol?
Hi. Let me go ahead and share my screen. Hi, everyone. My name is Marisol Vasquez, and I'm a senior majoring in biology in TAMIU in Laredo, Texas. Can you see my screen? It's loading.
Yes, we see it fine.
And I want to take the time to thank Dr. Wickenbach, Dr. Leggett, Dr. Gastel, and Abril for your continuous support during this program. The Science Influencers program has played a key role in both my academic and professional journey. So you can see my screen, correct?
Yes, it's fine.
Yes. Great, wonderful. So my research was a collaboration with the National Oceanic and Atmospheric Administration, where we accomplished the first urban heat island mapping campaign in Laredo, Texas. Laredo was one of 14 cities nationwide and the only city in Texas to do it this year. The importance of this study was not only the data collection, but also the public awareness of the heat-related deaths that have skyrocketed here in Laredo, Texas, going from 2 in 2021 and part in 22 to 15 in 2023. So the Calor campaign, short for Climate Action for Laredo to Organize Resilience, was a successful campaign to collect 47,000 temperatures and humidity data points across a 70.4 square mile radius in 9 routes. The environmental ground level data that was recorded was at a 1-second interval. And this was aimed to create high detailed quality maps of Laredo to take sustainable choices with city planning in South Texas. So the objectives, we wanted to map and collect the data for the heat distribution across residential, commercial, and industrial areas. We had 3 collection times, 6 to 7 a.m., 3 to 4, and 7 to 8 p.m. It was aimed to create high-resolution heat maps to inform city planning and public health, and engage the citizen scientists in a very unique community science event to understand the dangers of excessive heat, and as well as promoting environmental justice in the areas with limited green spaces that are exposed to much higher heat. The data collection was taking place where I'm actually presenting today at mile 1. And we assigned their starting points with the 9 routes. We collected the data in a 1-hour interval. They were trained. Our citizen scientists or volunteers were trained a week prior. We paid for their gas. We gave them stipends. We gave them a great energy here for participating in such an important and groundbreaking event. Each vehicle had a designated driver, a navigator who provided the directions of the route, and a note keeper to document any important observations during the collection time. The sensors were provided by Kappa Strategies. They are a third party who was hired by NOAA for the data analysis. And they recorded, like I said, temperature and humidity along with GPS in 1-second intervals. The diurnal temperature and the hyperlocal heat patterns were also recorded in those 3 collection times that I mentioned, 6 to 7, 3 to 4, and 7 to 8 p.m. For the data analysis, Kappa Strategies used a geospatial modeling process to create the high resolution temperature maps in 3 steps, which was step 1, download the filtered raw heat data. Then step 2, integrated multi-band land cover data from Centennial 2 satellites to calculate the vegetation and the surface features to identify the fine scale urban heat patterns. And step 3, it was a combined heat and land cover data in geostatistical model using machine learning to generate predictive surface maps. The study results demonstrated the concentration of hotspots in Laredo, specifically in industrial zones. Laredo is the #1 port of entry in the country, so we have a lot of traffic from semis and a very high concentration of concrete and asphalt. These areas consistently experience high temperatures. They do not cool down during the night, making them more dangerous, and because they retain and absorb the heat. These are the results. These are the 3 maps that were developed with the data collection thanks to our volunteers. I want to emphasize that in these maps, you do see a little bit of blue, but you do know that you can say that 98.3 is not cool at all. So it's not fully representative of the true heat with that blue there. And those blue pockets that you do see are a golf course, some parks, and dense vegetation. The data analysis, this also entailed 70% of the data was for model training, while the other 30 was reserved for the testing of the predictive accuracy. We really worked very hard to brand this campaign to make it exciting for the community, to create awareness, and to inform the public of the dangers of excessive heat. We aim to use electric vehicles. You see the cyber truck there with the sensor to create and keep a green mindset for the environment, for the sustainable future of Laredo. The model's performance was measured and adjusted with an R-squared value, indicating the strength of the predictions. And for the conclusions and applications, the Calor campaign produced high-resolution heat maps, identifying the heat spots in residential and commercial areas in Laredo. We also included our sister cities, Rio Bravo and El Cenizo, who are 2 small cities south of Laredo. And the data from these maps will guide city planners and developers to target climate strategies for vulnerable communities. This will also propose urban design strategies that will include reflective building materials, cool pavements, and to expand Laredo's tree canopies to reduce the heat, especially in marginalized areas. For the conclusion and applications, we're very excited that we have the data now. This is going to open doors for many grants that the city can apply for. Here in Laredo, it's very normal to see the heat come out of from the asphalt, and it's just something we're accustomed with, but it's not normal at all. And we're very excited that we collaborated with the city, RISC, the environmental nonprofit here in Laredo, to implement sustainable practices for a more resilient urban climate. And this is going to provide a baseline for the ongoing monitoring. We would love to participate again and be selected to do the campaign again. And I'm very grateful for the Science Influencers program. The ability to have a real life experience and do my internship over the summer at a environmental nonprofit RISC allowed me to really align myself and find a love that I did not know I had for environmental science. So very special thanks to RISC, the Rio Grande International Study Center, and my mentor, Martin Castro. He is the Watershed Science Director at RISC. And we hope that you check out the results. And if you have any questions about this logistical nightmare we had to do with over 100 volunteers, please reach out if you do have any. Thank you so much.
All right, thank you, Marisol. We certainly appreciate that and learning about your work down in Laredo over the summer. All right, so next up, we are going to go to Rhett Lewis, coming from Clemson University. And Rhett, if you're ready to go, Marisol, you have to remove your view. Okay, thank you. All right. Here's Rhett to talk about fermentation, analysis of the craft beverages. Excellent.
Good evening, everybody. So my name is Rhett Lewis. I'm an undergraduate senior here at Clemson University. I'm a food science major. And my project this summer was to kind of bring a fermentation lab to Clemson. Something super cool, something I never thought I'd be a part of, but very glad I was. Thank you to the Science Influencer program for that. But my whole focus with my abstract here, it's a little small. Can you guys see that or?
Yes, we see it fine.
Okay, perfect. So yeah, so my focus was to talk about a type of yeast. It's called Philly sour. And in this yeast, it's very advantageous because it helps to minimize contamination in brewing. So I was able to brew a type of sour beverage with this yeast in the new lab, and it was very fun. So besides obviously putting the lab together, I had a couple other objectives. So we had to use this lab for further investigation into microbiology, fermentation, everything that supports brewing. So we are now looking into a study for this fall about muscadine grapes. And with these grapes, we're going to see if there's any outside yeast, obviously there will be, on the grapes that could impact other types of our fermentation, because the craft brewing industry is exploding. Brewers obviously want consistent products. So that is our ultimate goal, is to try to see if, external yeasts will impact our finished product. So as you can kind of see in these pictures here in the results and discussion session, in this section, we have our hot side of the lab, which is like our brew station, something you would typically see in a commercial brewing setting. And you have your a hot liquor tank, mash tun, all that good stuff. And then on the left side, you have the cold side, which is going to be your cylindrical fermenters. That's obviously once you are done brewing the beer, the beers will be sitting in those tanks and fermenting after a few days. And just those pictures here. It's very short and sweet, but basically through this opportunity, got to bring forth a project to the fall. It was very cool. It really helped me learn more about how to interpret data, learning more about fermentation, microbiology, learned about different scientific articles. I'm very blessed that I was able to work under Dr. Paul Dawson and Alex Ryan Thompson. Alex is an extension officer here at Clemson, and Dr. Dawson is a full-time professor. And yeah, if there's any questions, I'm open to them. So thank you.
Well, thank you, Rhett. That was a very good chat. So we've heard about chickens and carrots and kombucha and now some craft brews. So I think we've got the menu just about covered there.
Absolutely.
Well done. Very good. Thank you again. All right, Sabrina Valdez.
Yeah.
Are you ready? Excellent. Sabrina also comes to us from Tarleton State University. And so let's hear about her research.
Rhett, can we get you to take down your presentation?
Yeah, it's not letting me share, I think. Or maybe you guys can see it
No,
now.
you should be there. You should be there, Sabrina.
Okay.
Yes,
That's
I can
it.
see
We
your
see it.
presentation.
Okay, perfect. Minimize. Well, hi everyone. My name is Sabrina. I am going to talk a little bit about myself first. So I just graduated with a bachelor's in animal science and pre-vet medicine. I've always wanted to become a veterinarian, but right before graduating, I started exploring research. And that's how I ended up at Charlton State University with the Bridging the Gap program. And currently, I'm in my first year of DVM here in Canada. So the program was in Stephenville, Texas at Charleston State University under the mentorship of Dr. Jessica Leatherwood. And some personal changes were that I gained a lot of confidence, especially when leading horses. They are very powerful, and you definitely have to have confidence when leading them. I became I gained a lot of adaptability skills and became aware of animal welfare. I always knew that I wanted to be an advocate of animal welfare, but didn't really know how. And this program definitely made me more aware of how to treat horses. And by educating myself, I was also able to, or am able to educate others. And I'm here with my research partner. I don't know if you guys can see my mouse, and I gained a lot of collaborative skills. I'm usually a kind of a perfectionist that likes to do everything because I think that I can do it better. But by working with others, I got to have other perspectives and a better outcome of the project. Also, we did not just collaborate with the animal science department at Charlton State. We also worked with the neuroscience department and the engineering department. So that was really cool to see other perspectives. I also gained a lot of knowledge with research methods and the importance of statistics, and also gained skills on literature review. So I'm going to be talking about our project, which was exploring the use of surface electroencephalography and the identification of lameness in mature horses, which is basically utilizing a device, which is this one right here, to be able to detect lameness. Lameness is a clinical sign that may affect horses from all ages or all kinds of breeds. It is an abnormal gait, often caused by pain, and the current gold standard for measuring it is the American Association of Equine Practitioners lameness scale. It is quite subjective as it's a human evaluating the horse, and it depends on the experience that person has to be able to detect the lameness. And it also depends on not just the subjectiveness, but we also wanted to find out if we could find a tool to detect lameness earlier and avoid chronic cases. So that's where surface EEG comes in. It's potentially measures the wavelengths or different frequencies emitted by the brain, and high frequency bands when seen in a resting horse could potentially indicate pain, and this tool has been used in other species. We also use blood biomarkers. In my case, I studied substance P, which is a neuropeptide linked to pain, and we just wanted to know if there was a correlation between the surface EEG bands and the blood biomarker. So the objectives basically were to determine if there was an effect of time while of the conducting a lameness evaluation on a substance P, which is the blood biomarker, and the frequency bands of the EEG. So here we place the cap on a standard position for all horses. Then we process the frequencies and
filtered them to divide them into the 5 different kinds of bands. So high frequency bands would be from gamma to alpha, and low frequency bands will be from theta to delta. A resting horse would show only theta and delta, and a horse that is more active would show high frequency bands. So again, if you see high frequency bands on a horse that is resting, that could be indicative of pain. This is the process of taking the substance P sample from the serum. And this is the lameness test that was done in between of pre and post samples. So some key findings were that there was no effect of time on substance P, and we found a negative relationship between age and substance P, and that is accurate with other literature. As you age, substance P lowers. And there was a moderate to strong relationship between age and lameness, and no relationship between substance P and lameness, and no effect of time on the frequency bands. So elements for future career, I gained a lot of networking skills, built up my community all throughout the US. We flew to North Dakota, also not just stayed in Stephenville, but we also helped with projects up in Amarillo, Texas, and also down to College Station. I gained a lot of skills for literature review and also equine and hands-on exposure that boosted up my horsemanship skills and became more aware of the animal welfare and ethical considerations. So thank you to my amazing mentor, Dr. Jessica Leatherwood, my partner, Jesus Cortez, and everyone involved in this project. Thank you.
Fantastic. And great photo finish there too. Sabrina. All right, let's go ahead and open up if we can see if there's any questions from the floor. No. And I think we can you can turn on your cameras if you would like, and turn on your microphones and ask questions. Maybe some of the students have questions for each other. Also, we have a few participants on who are looking to become the next cohort members of REU programs. So for our current REU students, what advice would you give to other undergraduates who seek the same experience that you had?
Can I jump in, Dr. Wickenbach?
Yeah, sure.
I would really suggest to embrace the learning curve, because you're going from one field to the next, from academic to professional. So embrace the learning curve. Don't beat yourself up, because it can be a very long curve. And get yourself a notebook and write everything down. Get a journal so you can practice the post-reflective exercises, and just constantly document as much as you can. It'll be very helpful.
That's great advice, actually. I need to get a couple of notebooks myself. Helps out with the memory. Other advice?
If I could chime in, Dr. Wikibatch, I think that going off of what she just said, is very important to embrace the learning. And one thing that I really took forward was embracing that my professors, these are people that have gone through everything that I dream of doing, and they have already walked the path and are just full of knowledge. And I feel that just talking to them on a regular basis and just getting to know them on a deeper connection besides just professor and student, talking to them just as a normal person, I feel like goes a long way. I feel like just treat your professors the same way you would treat anybody else.
Mhm. I want to note Sabrina and Jesus, I think you both got to go to North Dakota, you said? And how long were you
Yes.
there?
So we spent the majority of the time in Texas, but at the end, we actually met with the other students of REU and did the final presentation in North Dakota. And that was around 3 days. Maybe we
Oh, okay.
arrived, then presented, and then left. But it was actually an amazing experience, and we got to see the Blue Angels as we arrived. And I just thought of the movie. What is the name of the
Top Gun, perhaps?
Yeah, Top Gun. I can't believe I just forgot it. But yeah.
Oh, that's fantastic. And so I'm really amazed at the range of experiences that everybody had from North Dakota to Mexico to the Northeast to all the way to the southern border. So those are fantastic opportunities. We really hope that the experiences you gained over this past summer will stay with you and encourage you to continue, as Sabrina is doing right now up in Canada, continue seeking out that path to become a dedicated scientist for the betterment of all of us. And we really appreciate, again, all of your contributions. Thank you for participating in the third annual research symposium. Abril has her hand up.
Yes,
Go ahead.
I just wanted to take this moment and take advantage of that some of the REU students are here and ask you guys if you would like me to showcase all your amazing experiences. You can email me with either like a little text of your experience or just like pictures of you in the lab so that I can post that on the news, social media and everything. And, everybody can know about what you did and want to encourage them to also gain some experience in the field. And on the webpage of the REU, I don't know if you guys ever visited that, www.reu.org. I can put it on the Q&A, I guess. Yes. There is a form that is called Share News with Us, and you can upload all of that there if you'd like me to post it. So just throwing in that opportunity.
Oh, yeah, excellent point. And that goes for all programs, no matter where they're located. Please do share your stories. I know Caleb has a pretty active following on his social channel, and so we would like to help promote those. So, Dr. Gastel, any parting words?
Yeah, I just wanted to compliment everybody. It was fascinating to learn from everybody's research and hear more broadly about your experience and sort of see how people grew and developed during the summer. So I'm very impressed and want to congratulate everybody and wish you all the best in your continuing endeavors.
Absolutely. I feel the same way. Thanks again, everybody, for joining us. And wishing you all the best for a fine end of the semester and an enjoyable fall.
Thank you so much.
All right. Bye-bye.
Thank you.
Thank you.
Bye. Thank you. Have a great day.
Thank you.
Yeah, thank you so much.